The world production of apple is approximately 89 million tons, and China, USA, Russia, Germany, Japan are the leader producers and exporters. In Pakistan, the production is around 590,000 tons, and in Gilgit-Baltistan, my study area, the production is around 370,000 tons, which holds second position in the production among other fruits in the region. Local varieties is known by name and very poorly studied. There are threats to the germplasm in the region as farmers are preferring the new and commercial varieties. Therefore, traditional and local varieties are undervalued and replaced by the commercial ones. However, these local varieties could be often demonstrates the resistance to biotic and abiotic stresses, for instance, against drought and apple scab and powdery mildew caused by the fungal pathogens, Venturia inequalis and Photosphaera leucoteria, respectively. And so in GB, it provides a habitat value and the second important fruit crop after apricot. However, very less to no scientific evidence available about the local germplasm. Local varieties, because of having low yielding and less appealing appearance, are being replaced by the modern varieties, which are sweeter, low bitterness or sourness, and large size and homogeneous color and appearance. Moreover, they have high market demands and production. Currently, promotion of modern varieties causing narrowing of the local gene pool in the region. So to analyze this influence and replacement effect on local varietal diversity, we propose a hypothesis that varietal richness and diversity is similar across location, but declines from very remote to market close areas. And for this purpose, we selected two dead-end valleys, and each valley, we have for one valley, we have 4 villages in Baltistan region, and the other valley from Gilgit region, we have 3 villages, and each valley is accessible only from one side, and hence did probably experience introduction of plant genetic material only from its lower sides of both valleys. And this is the Karakoram Highway, which is currently also utilizing a new road between China and Pakistan, the currently CPEC road. Exhaustive snowball sampling was done, and minimum of 5 households per village were identified having owned local varieties. Farmers were interviewed about the management history usage of individual tree, and every variety and every individual per variety per household was sampled until a new variety was identified or at least considered morphologically different from all sampled specimens so far. In addition, we also collected samples from the government nurseries of the local varieties as a reference material, making a total of 106 individual tree samples. Detailed fruit morphological description and measurement was done using predefined fruit descriptors. Varietal diversity was estimated from the abundance data. SSR markers were screened out from the literature review, and also Laboratory of Conservation Biology at University of Marburg were contacted to find out a common set of markers. But pre-screening suggested that this is rather difficult as the same set is not polymorphic in our tested sample. So we finalized 11 highly polymorphic SSR markers for further analysis. So from our study, we observed that there are no arboricultural activities in the region. And we found around 14 to 17 meter tall tree with 114 to 171 centimeter stem diameter. The fruit morphological diversity was also observed. Here are some examples. The local name of this variety is Ali Khan Balt. It has an irregular fruit shape with rough surface, but sweet in taste. This is another variety named Desi Loopmoor. It has a crooked fruit from both calyx and petiole side. Also, it has 5 edged fruits. The local name of this variety is Zakushu. It has a very long petiole and it is very sour in taste, suitable for wine production, but unfortunately the wine production is prohibited in the Gilgit region. The local name of this variety is Markushu. It has a very strong red coat color. Interestingly, we also found and identified the wild relatives of apple, Malus baccata, in the region, in one of the villages, Bisal village, which is the dead end village of one of the valley. The local name of this apple is Kuju. It has a very small fruit of 1 centimeter and very sour in taste. So considering this morphological diversity, we also estimated varietal diversity based on the abundance data of each variety and number of varieties found in each location. There are two different trends looking at, there are two different trends looking at the alpha diversity, that is diversity present in each village. That diversity is decreasing from villages from the Gilgit region from very remote to the market close villages, whereas there is an opposite trend in Gilgit region villages. This might be due to the fact that Gilgit region is more near to the CPEC road and the region is more developed as compared to the Baltistan region, and there is more faster replacement of the older varieties from the modern ones. While looking at the beta diversity, which is on the node of this dendrogram, showing the diversity among villages, represents that Gilgit villages are more diverse from each other, which means they are sharing less common varieties as compared to the Baltistan region. They are more diverse, they have more varieties, but more of them are commonly sharing among different villages of Baltistan region, whereas in opposite in Gilgit region. So we also verified the farming naming of the varieties through SSR-based DNA fingerprinting, because region is rich in varieties exerting the same local names, homonyms, and some varieties with different local names, synonyms. And we found 36% true to type varieties, which means they have same name and same genotypes, whereas tree owners named about 51% of the varieties incorrectly into putative homonyms, means they said in that it's the same name, but it has different genotypes according to the genetic analysis, and putative synonyms and ambiguous genotypes behaving both homonyms and synonyms characters. There are several reasons of this misnaming. Number one, SGB has linguistic differences across ethnicities. Number two, farmers are unaware and having lack of interest. Number three, there is incorrect determination or inheritance, as well as somatic mutation that may lead to the marked phenotypic differences, but not genotypic differences. We also performed the tanglogram analysis between genetic dendrogram and phenotypic dendrogram. And interestingly, we also found a good entanglement quality and correlation of 0.62, which shows that these two dendrograms are statistically moderately similar. This might be the sign of genetically linked trait, which merits further in-depth studies. So we concluded that there is a large pool of still untapped varietal diversity in apple, but there is an urgent need for conservation as we discovered ongoing process of loss of diversity. There is a need to establish a labeling system of available germplasm in the region according to their specific fruit and tree characteristics. Most of the varieties were characterized by low yields but with hardly marketable traits. But their genetic setup could be valuable for future crop improvement. For future repetition of such research may include the modern varieties, collection from the farmers and government nurseries along with the local primitive ones. So thank you very much for your attention.
Yeah, thank you very much, Mr. Aslan, for this very clear. I thought it was very well done and very informative, and also nice pictures, making me feel like eating an apple.
Thank you.
Now, I don't see any questions in the question and answer side. I wonder whether someone from our group here in the Zoom room has a question.
Mm-hmm.
If not, my first question would be, are those apple varieties already conserved somewhere in situ or ex situ? Or would that also be something to focus on in the near future in order not to lose those varieties?
Yes, actually, that is not much conserved, but as we also collected the samples from the nurseries of these local varieties, to have we also looked at how much of these local varieties are conserved or maintained in the nurseries. But unfortunately, we found around 30 to 40% varieties in the nurseries, and rest are only in the farmer field. And even in the farmer field, it is abundant, not very much cared, maybe because they are more remote areas and they are just kept for the animal feed and like this.
In Germany, in some places, we have those gardens where people try to conserve all the traditional
Yes.
Tree varieties. Could you imagine something similar to happen also in Pakistan?
Yes, this is what we are also doing research on, and we are also thinking to make a collaboration with the research institutes in the local research institute of that Gilgit-Baltistan region. But the first step is to characterize the available germplasm and to see that which varieties are more endangered and need to be conserved first, and then further. Yeah.
And would you conserve the seed of those trees, or more like in situ growing the trees, or maybe both?
I think both will be the option, but in field conservation is also more good that we have. Like in Germany, I also found there are more trees growing in the farms. They have, yeah, they have different pomologian societies which are conserving in Germany. The concept is very nice.
Yeah.
That concept has to be also introduced in other countries like in Pakistan.
That would be really great, yeah. Getting people excited about the diversity which they can cultivate. That would be really excellent.
Yeah.
So are there more questions, maybe from the other participants in our Zoom room? Question? If not, well, I think time is also over for this presentation.
Okay.
Still, the second presenter from Kenya is us, which is unfortunate, but well, I can't change it. It would have been a presentation on chickpea tolerance to soil acidity, which would have been very interesting. Unfortunately, he's not in. So we would move to the third presentation to be given by Joshua Okonya from Uganda. delivery of clean planting material of orange fleshed sweet potato to smallholder farmers through decentralized Uganda. Joshua, please start sharing your screen and start your presentation.
Thank you. Thank you, Bettina, and good morning to everyone in this session. So, Joshua Kwanya is my name.
Could you put it in, please?
Yeah.
Thank you.
Yeah, so Joshua Okonya is my name, and I'm based in Uganda. I would like to talk about the delivery of green planting material of the orange fresh sweet potato varieties. This research was done in Uganda by myself, Norman Pikiriza, Dr. Fred Brandt, Joyce Maru, and Simon Heck. So they outlined a bit of the background problem statement, what we want to achieve, where we are working, our approach, what findings we got, and the way forward. So a bit of the background is that about the importance of vitamin A. Sorry. Yeah, that about the project. This is part of this work was done under the consortium of the development and delivery of biofortified crops at scale, JDBio, which is a project that is working in 5 countries: Bangladesh, Kenya, Uganda, and Nigeria and Ethiopia. And in Uganda, we are working in 14 districts spread across the country. So crop biofortification is one of the most promising approaches that has been shown to reduce malnutrition in the recent years, and this is mainly in the resource poor communities that cannot afford the expensive vitamin A food sources like liver, fish. And we find that in these communities, the vitamin A-rich orange fresh sweet potato has proven to be quite effective in improving the health of children, especially those ones under the years of 2, pregnant women, breastfeeding mothers. And this comes in communities where we work, where we have quite a high prevalence of vitamin A deficiency that is up to 38%. So vitamin A, how important is vitamin A to our bodies? We notice that it's an essential micronutrient for the immune system. It plays an important role in maintaining epithelial tissues. It's severe. Yeah, vitamin A deficiency can cause eye damage and can lead to blindness, especially in children. It increases severity to infections such as measles and diarrhea, and it's common in dry environments where fruits and vegetables are scarce. And these are the areas where we are working. Talking about vitamin A deficiency and boosting immune system, and we notice that of late we have the COVID-19, which is also a virus, and we're not sure yet. So we find that having a good immune system is really important. So the problem, what is the problem? The problem we want to address is that there is limited access to improved and clean orange fresh sweet potato varieties. And this one has limited the realization of the full potential of this crop or of these varieties. What are some of the factors that have contributed to this? There is low sensitization or little investment in the marketing of clean planting material. These are mainly at the moment we only have 2 tissue culture laboratories in the whole country that produce and multiply clean sweet potato or tissue culture or vegetatively propagated crops. And by the decentralized vine multipliers, or what we call DVMs, and basically there are not many traditional vine multipliers. So prolonged dry seasons where farmers lose all the vines, and this is also contributed by the lack of knowledge of how to conserve vines over a dry period. there is quite a lack or the high cost of irrigation involved, and this affects the production, especially in the first season where vines are not available and there's low demand, yeah, and of these vitamin A rich varieties. And this is mainly by, it's more or less of project driven than being incorporated into the culture of people. Normally people here are not used to buying sweet potato planting material because they usually pick it free from their neighbors. Whereas for the orange fresh sweet potato varieties, they need to be bought, and anyone who multiplies them cannot give them out for free. So what does this project want to achieve? We want to increase the availability of disease-free and high-yielding planting material for the orange fresh sweet potato varieties. And we do this by carefully identifying, selecting farmers with potential to become seed entrepreneurs. We recruit them. We train them in rapid vine multiplication techniques and doing farming as a business. We support them with the initial planting material, sometimes also with fencing material, irrigation equipment. And then we promote, we go ahead to promote. Now that once we are sure the production is there, we go ahead to promote the orange fresh potato in the varieties. And we do this in schools, in the local governments, in the health centers, so that we can create demand. So we work, like I said, in 14 districts in Uganda, and you can see the districts are scattered mainly in the northern areas bordering South Sudan, where we have a large number of refugees influxing from South Sudan into Uganda. That's why we target mainly the northern districts. And then the Acholi region is also partly affected by the, it has been over I think nearly 30 years. There was the road resistance army, Joseph Kony, and these people were mainly displaced and they were living in camps, so we have high malnutrition rates. And then the Karamoja region, Moroto and Kotido, where we have the cattle, high cases of cattle rustlers and nomads, and this place is a bit arid. Poverty is highest in Uganda, just like illiteracy. And also the Buked region, the eastern Uganda, there are about 4 districts which have high poverty and malnutrition rates. And this is a bit of the pictures of the places where we work, but you can really see that the environment is quite arid. They live in temporary shelters. They don't have, they don't have clean water. Some of them harvest rainwater from tree trunks. And then also these same places are continuously being hit by catastrophes like the desert locust that invaded in the last year and this year in all the districts that are bordering Kenya. So we do selection. We have a criteria that we use to select a good decentralized vine multiplier, DVM, and these ones should be a progressive farmer, should be a seed entrepreneur, must have a permanent water source, must have a business acumen, should be able to demonstrate that they will continue in business after the project has ended. They should be able to invest in fencing material. They should be able to read and write so that they can keep records. They should be centrally located so that the vines are accessible, because accessibility is one of the ways that one of the factors that has reduced the adoption rate. And then we should have a telephone number, living in areas with a cellular coverage that they can be reached. They should own land and must be permanent residents. So our approach is that we do train, we recruit farmers. Those farmers, we train them in rapid vine multiplication. We give them planting material and they planted. You can see pictures of the sweet potato fields that under multiplication, and these are multiplying for vine production, not for root. We do train in good agronomic practices, and we also conduct nutrition trainings to improve the nutrition education, basically so that people can understand the importance of having a balanced diet and the need to incorporate vitamin A rich food into their diets. And then we do distribute vines, and I think currently distributed to nearly 4 million households that have received. Yeah, 4 million cartons, and these have been distributed over 12,000. And this was only for the first season. So what are some of the findings we got? We noticed that the perception of the stakeholders on how we can increase the consumption of the orange fresh sweet potato varieties. And we see here that we need to ensure that there are enough quantities of OFSP vines that are produced in each sub-county. People are saying the vines are very far, so they need to have nearly a vine multiplier in each, or in a not longer than a 10-kilometer radius. Otherwise, they find it expensive to go and collect them. We need more rigorous marketing through especially radio to show market base. And if we have stands with the orange fresh sweet potato, we should label them and people know. That's why I can find the vitamin A rich crop or roots, not the white fresh crop. And then we need to do community sensitization and nutritional benefits. Yeah, because many people are religious here, so they believe we can go through religious institutions like churches or through social gatherings where local leaders can speak about the orange fresh sweet potato varieties. This would increase consumption of OFSP. Yeah, we should do the overview that we should set up demo plots at the sub-county headquarters, FA units, schools, and also at demo farmers fields. So they also the overview that we do training in value addition and processing of OFSP. This would include cooking demos and different recipes that can where OFSP can be used, puree, or also to include the OFSP in bakery products, something that has already taken on. And you can find many breads and bakery products on the market, cookies, which have been made with about 30 to 40% of sweet potato, replacing wheat. So about the challenges that challenges involved in the production of OFSP. Yeah, stakeholders mentioned that the varieties are not as hard, meaning that they have little dry matter. Yeah, so they cannot easily be good. They say the varieties also at the beginning of the rain season are not there, yet they cannot do irrigate. They say that many farmers in certain districts have never heard about these ones, so we need to increase our advocacy. They say that many people are not aware of the nutritional benefits, so they just eat for the sake, not eating nutritionally. Yeah, the varieties are a bit soft. I've mentioned that because of low dry matter compared to the traditional ones, which are white and quite have high dry matter and are hard. And then they say that some varieties are equally susceptible, including the orange fresh varieties. And then they don't stay long after maturity. So when we asked about the opportunities of producing, market, and consuming OFSP, they say, okay, the opportunities are quite there. The orange color is very attractive, yeah, and especially to children, they do like it compared to the white flesh. They say the varieties mature early, 3 to 4 months. To traditional ones take about 4 to 6 months to mature. They say it fetches quite a lot of money when you're selling the vines to the NGOs. Yeah, and they say you can use this as an ingredient in cakes, mandazi, chapati, and that is which you cannot do when you are having white fresh varieties. Yeah, and they say because this variety is soft, even if you don't have sauce, you can take it comfortably without tea or water, without the fear of choking, because normally it's eaten as a snack for breakfast. And the way forward, we notice that there is need for to intensify the marketing and awareness creation so that we can achieve the scale that we want with these orange fresh sweet potato varieties. We also notice that we need to train more vine multipliers and support them to be certified so that they can stay in business. We need to prepare new vine multipliers to join existing associations that they can increase their network. We need to engage health assistants and the village health team members to popularize the varieties. And we need to support demos at schools and sub-county offices and the health centers. Yeah, this project has a number of partners in the country. We are working with the Ministry of Agriculture, with the World Food Program, Harvest Plus, the National Crop Resources Research Institute, the Rural World Federation, MASECO, and these are mainly all non-governmental organizations and relief agencies, research organizations as well. It's being funded by GFID in the UK, and also work with a number of wildlife associations. Thank you.
Okay, thank you very much, Joshua, for this very clear and beautiful presentation. It was also very informative. I'm very happy to announce that Richard is now also online. Welcome, Richard.
Thank you very much.
Accommodate your call, but first let us discuss a little bit the presentation of Joshua. We still have enough time for this session. So I'm checking the question and answer side, but there are no questions there. Maybe while you are thinking about other questions, I would like to ask Joshua, what are the challenges when multiplying this orange-fleshed sweet potato in a decentralized manner, and how do you assure quality control?
Yeah, thank you. Thank you, Bettina. I think the challenges mainly for vine multipliers has been the being able to multiply during the dry season. So irrigation. So a vine multiplier must have access to either permanent water source, and some places like where we work in the arid areas, there is no water source. So people rely on water that has been dug and pumped in the pump that supply the communities. So that has been one of the major challenges. But for farmers who have access to, say, a swamp or a shadoof or a well, a shallow well, then for them, they are able to do the multiplication during the dry season. So in that case, multiplication becomes quite easy. Also, maybe another challenge is that during the dry season, when multiplication is happening, you find that these are pastoralist communities. You must be able to fence off your field. And fencing material is quite expensive. So for those farmers who are not able to fence, then you find that animals usually damage and eat the crop, and then they cannot be in business for long.
And what about virus diseases?
Sorry?
What about virus diseases?
Yeah, so for the quality control, we do, for every vine multiplier, we have rules that we set for them. If we are to support them, we normally source for them or support them, subsidize the cost of the initial planting material by 50%. So you find that from the tissue culture company that we have in Kampala, 1,000 cuttings go for 40,000 shillings, which is about $12. So our project subsidizes the cost to about half, about $6, and then we supply them with clean, which is virus-free planting material. So it's from that material that they go and multiply, and we encourage them that in high pressure, in areas with high virus pressure, after every 4 generations, they must replace their seed. Whereas in areas with the low virus pressure, after every 6 generations, they must replace their seed. So that's how we try to ensure that quality is maintained and viruses are kept out of the seed multiplication plots for the decentralized vine multipliers. Yeah, okay. But yeah, and then we also don't allow them to source from other farmers. We have an agreement with them, and we clearly stipulate you are not allowed to source material where you don't know the origin. So you must know the origin of the material that you are going to supply. So that's how we try to.
Education is, of course, a good step.
To do some checks.
Further questions from the audience? If not, I would like to ask, do you think that selling to NGOs is a long-term market for those multipliers?
Sorry, I'm selling to NGOs. Is it a long-term market? I missed the question.
Yeah, whether this is a sustainable market. I mean, NGOs, they have projects, they are there for 3 years and then go away. Is that really a sustainable market for the multipliers, or would it not be even required to have a really local market?
Yes, absolutely not. Absolutely not. And selling to NGOs is not sustainable at all, because these are projects. They come after 2 years, 3 years, maximum 5 years, they are gone. So we encourage that the vine multiplier must have a business acumen. They should be able to do local marketing. They should be able to market themselves. So otherwise, they will die out at the end of the project. So we encourage them that they should, because in some areas, especially these drought-prone areas, also we have the traditional vine multipliers. These normally sell the local varieties of sweet potato. So we try to encourage them to liaise, or we try to recruit the already existing traditional multipliers, because for them they do that as a business usually. So we try to bring those on board, because they know where the communities around them know where they are located, and then they have access to permanent water. So if they really don't rely on projects, they sell to the general population. So we encourage that they should do, for example, radio shows. They should go to schools. They should reach out to health centers, and there is in there they are able to find that they have continuous market for their vines even at the end of the project. But I agree, NGOs are not, the market for NGOs is not sustainable.
I really like your approach. This means involving young people, sensitizing them, and this could also really translate into better nutrition of those school kids at the end. So congratulations to this approach. I think right now we move to the next presentation, which would be given by Richard. Richard Onwonga, increasing chickpea tolerance to soil acidity, screening land races for tolerance to aluminium and manganese toxicity in solution culture. So Richard, if you could share your screen.
Okay, fine. Let me share the screen. Thanks very much, Bettina and colleagues. Sorry, I missed part of the presentations, but I hope I'll be able to listen to the next ones after this one. Let me share my screen. Hope this is loaded. So I hope you can be able to see my screen. That is the title of my presentation.
Mode.
Just as our facilitator has indicated, I don't know why it is not going to the presentation mode. My goodness. Sorry, for strange reasons, it doesn't want to get the presentation mode, but I'll keep trying. So the title of my presentation is Increasing Chickpea Tolerance to Soil Acidity, and this screening land races for tolerance to aluminium and manganese toxicity in solution culture. This is a joint effort between myself, then we have Katka, Paradip, Wendy Fancy, and Richard Bear. I'm from the University of Nairobi, Department of Land Resource Management and Agricultural Technology, whereas my colleagues are from Murdoch University, College of Science, Health, Engineering, and they are from Australia. So this is how I'm going to present my study very quickly. I'll give the background, then study design, and obviously conclusions and recommendations. So then to get straight to the presentation, and mainly why I'm focusing on chickpea. It is documented that chickpea is one of the founder crops of modern agriculture and is an economically important crop that is grown globally. Some of the key aspects in terms of the importance of chickpea is in terms of carbohydrates. It gives a very high level of carbohydrates, or rather it has proteins, vitamins, and minerals, and also it contributes to enhancing soil fertility in terms of biological nitrogen fixation. Some things that are also known about chickpea is that it's a low input crop, and in fact it does well in marginal areas with very low rainfall. The residues can be used to improve soil fertility, and particularly in Kenya, chickpea has been recommended as one of the adaptation crops to climate change, and that is according to the Ministry of Agriculture. Then chickpea has some production constraints, key among them being abiotic stresses, pests and diseases. Then we have biotic stresses, which are responsible for up to 48% of chickpea yield losses. And these include, among others, cold stress. We have heat stress, we have waterlogging, and the rest. But my focus for this particular presentation will be on soil acidity as one of the biotic stresses that affect chickpea production. So soil acidity is known to hamper crop production and is responsible for a reduction in yields for up to 70%. So what soil acidity does, it enhances the levels of magnesium and manganese to be very high in the soil solution, and they affect the uptake of other nutrients. So the toxic effects of magnesium are considered to precede those of aluminum in the process of acidification. And this will become clear as we move on. So some of the reasons why I had to undertake this study is that most research that has been done on chickpea, the focus on soil acidity is limited, but other aspects, for example, increasing yield, tolerance to salinity, waterlogging, and so on has been done. But there's very little that has been done in regard to soil acidity. But again, we know that breeding for tolerance to acidity, that's aluminum and magnesium toxicity, is the starting point for any breeding program. And therefore, it is important that we start with screening of chickpea, and especially the landlesses and wild relatives, to find out which ones could be tolerant to aluminum and magnesium toxicity, and thereafter they can be taken to the field for further breeding programs. So I don't think I will have to discuss much on this, but it's important to mention that the screening took advantage of the protocols that have been developed at Murdoch University to screen large accessions of chickpea that have been collected from various parts of the world. And the reason for this is that we anticipate to identify GKP accessions or varieties that could be tolerant to the acidic conditions that are prevalent in both Australia and also in East Africa. So what we need to know is that the screening was done in a growth chamber. The study was run in 2 phases, implying that the chickpea landlasses was screened for aluminum first, and then the second phase, we had the same landlasses being screened for manganese toxicity. This may not be visible, but in general, we have various chickpea accessions or landlasses that have been obtained from various parts of the world. And notably, we can talk of India, Ethiopia, Bangladesh. We have some as far as from Nepal and a few from Australia and Bangladesh. So this, I don't need to spend a lot of time on this because these are the usual preparation of seeds procedures for screening. So we have to start with the seeds clarification. Then we move to seed sterilization. Then we have pre-germination. And then the preparation of the nutrient solutions where we are going to grow the seeds. And this is what we have here for aluminum and for testing for toxicity or tolerance of the accessions to aluminum toxicity. The main solution containing the aluminum we used was aluminum chloride, and for magnesium was magnesium sulfate. The experimental design was a computer randomized design with a split plot arrangement where the main plots were the 3 levels of aluminum. I used 0, 15, and 60 micrometers of aluminum. And then for manganese, 2 levels were used. That is 2 and 150 micromillimolars. And then planting of pre-germinated seeds was done in the growth room. For the aluminum, the seeds were pre-germinated for 4 days and then taken to the growth container that it was containing the solution. The same procedure was followed in manganese. The only difference is that the pre-germinated seeds were subjected to 7 days of manganese treatment. That is the 2 micromillimeter level. And then the experiment was run for 26 days, whereas for aluminum, the experiment was run for 10 days, plus for the 4 days of the pre-germination. And then a few procedures were followed in the growth chamber, where the pH was being measured almost on a daily basis and maintained at 4.2 for aluminum and also 5.2 for manganese. And then the solution was also being sampled on a daily basis for determination of the levels of aluminum and other nutrients. So this is the extraction of the solution going on there. And then after this, at harvest of the seedlings, various parameters were being measured, where we had the root length that was measured prior to planting and after at harvest. That is 10 days for aluminum, and as I mentioned for manganese was after 26 days. Then we had the root and shoot dry weight also being determined for both the accessions under aluminium and chickpea. Sorry, aluminium and manganese toxicity assessment. So for manganese, toxicity symptoms were being scored, and we had failure scales that is 0 to 5, where 0, the plant did not show any symptoms, and 5, the plant had symptoms, and from the diagram it can be seen to be really affected. And the description of what I was looking for is also indicated. The same parameters were measured for aluminium for manganese, just as for aluminium, that is the root and shoot dry matter, not forgetting the shoot length and also the root length. So for the dry weight determination, this was done in the oven for 4 days at 60 degrees centigrade. And then the weights measured using a weighing parents. Then for it to be easy to classify the accessions, as we have seen, there were many over 100. Some indices were developed, and one of them is the change in root length. We had also the relative root elongation. Root tolerance index was also calculated. We had also the relative root and shoot dry weight, and also some measurements were done with respect to toxicity symptoms. So this was very key to make it easy for the classification of the various accessions. So that is how mentioned for manganese, the toxic symptoms. Then after this analysis, some classification was done. It is hidden here, but I can indicate that depending on these indices, any accession that was found to be above 70%, for example, in terms of the relative root elongation or relative roots and shoot dry weight, it was considered to be tolerant to aluminium toxicity. It was between 69 and 50, then it was classified as moderate, and it was below 50%. It was classified as being sensitive. So the same was done for manganese toxicity. The only different criteria that was applied for the toxicity symptoms, where it was indicated that if the symptom score was above 3.5, then that accession is sensitive. If it's between 3.5 and 2.5, then it was moderate. And if it is below 2.5, the accession was classified as being sensitive. Statistical analysis was done using a two-way ANOVA, and means was separated using the risk significant difference. Correlations of the various parameters measured was also done. And these are the results. So using the relative elongation index, over 85% of the accessions evaluated were found to be sensitive at both the 15 and the 60 micromilliters of aluminium concentrations. And only about 15% of the same were tolerant, but only at the 15 aluminium concentration level, and sensitive at the 60 aluminium concentration level. And the accessions are indicated, and the origin can be traced from table 1 that I presented, but these were the accessions that were found to be tolerant in the 15 aluminium concentration level, and their origin is as indicated. Most of them actually were from Ethiopia, and one from Bangladesh, and some were belonging to the wild Caesar. So this is what we had in terms of the tolerance, and this is a grouping of how the accessions are spread using a scatter diagram, but basically it is as I presented them here. Then looking at the dry weights, the classification that was used is the same as for the relative root elongation. So based on this, the accessions, I don't know how to expand this because some information is hidden.
Richard, I also need to ask you to hurry up a little bit because the presentation time was you had those 10 minutes already. So please summarize your results and conclusions.
Yeah, so these are the results. The most important results I have to, sorry, I didn't take note of the time. So this is what we can summarize. In terms of magnesium toxicity, we had the following. 32 accessions could actually be classified as being tolerant, and this is 49.24 and 41.054. The rest was sensitive with respect to manganese toxicity. So in conclusion, this is what I have to say. If I may rush to the conclusion, that through this, we were able to identify one chickpea accession that was actually tolerant to both aluminium and manganese, and that is accession 49938. Two were identified as being moderately tolerant to aluminium and manganese, 41060 and 49298, and the rest appeared to be sensitive across the two toxicities that were being investigated, that is aluminium and manganese. So from the results we have already obtained, there's high possibility of isolating land races that are both tolerant to aluminium and magnesium toxicity. And I would like to acknowledge my collaborators and also to mention that this is an ongoing study that is being supported by the Australian Award Program. Thank you very much, and sorry for taking a lot of time.
Yeah, no problem. I think we can still manage. Thank you very much for this presentation. I wonder, are there any questions from the group? I don't see a question in the question and answer chat. Is there someone from the group who would like to ask a question? If not, maybe I ask one question. As I'm a plant breeder by training, I would like to know whether you estimated the heritability of those tolerance indices in the hydroponic. So whether you have an estimate of the genetic correlation of this results from the screening in the hydroponic system and performance under field conditions.
Yes, I would like to say that we have not yet done this. This is basically preliminary screening, and I'm thinking once the best accessions in terms of tolerance, manganese, and aluminium toxicity are identified, then we can move to the next level. Because the idea after this is to take the best, the elite land races to the field for further breeding, and then I think people like you now will come in. Genetists will come in and assist in terms of looking at other parameters that are key in actually ensuring that these accessions can be progressed and eventually taken to the farmers by virtue of the fact that they are qualified to be tolerant to manganese and aluminium toxicity. So at this level, not yet. That one has not been done.
Okay.
Yes. And
are the wild tolerant accessions very similar to the cultivated accessions? Will it be easy to use them in a
Among the cultivated accessions that we're using, we had Srika and Amber, but they were not found to be tolerant manganese and aluminum toxicity. So that's why we are more or less biased to the landlasses and also to the wild Caesar. They have shown some promise as compared to the cultivated varieties.
Okay. Well, then I wish you good luck for the continuation of this study, which is very important. Acid soil is a big constraint in many parts of Africa. And I suggest that we now move on to the fourth presentation.
Okay.
Yamin Humaydan, in vitro induced polyploidy in Celozia argentea variety. So Richard, you need to stop sharing, and Mr. Yamin, please start sharing your screen.
Good afternoon, everybody.
Okay.
My name is Yamin Shmait. I am from Lebanon, and I'm currently doing my PhD studies at the Czech University of Life Sciences in Prague at the Faculty of Tropical Agri-Sciences. Please allow me to introduce this research titled In Vitro Induced Polyploidy in Celozia argentea variety plumosa. Several other researchers and myself participated in this work. Celozia argentea is an annual herb of tropical origin. It belongs to the Amaranthaceae family. The generic name Celozia is derived from ancient Greek language, meaning burning, and that refers to the flower heads. As you can see, the inflorescence in the figure 1, part A and B. Part C on the right, we look at the seeds of Celozia. These are harvested in autumn when the infructescence matures. Then the ripened dried seeds are used in traditional medicine in several Asian countries, especially in China. Recently, there is a pharmacological interest in the components of Celozia, where several compounds like saponins, alkaloids, and phenols have been isolated and studied for their use for several illnesses like tumors, fever, and diabetes, and several other diseases as well. The main effort of breeders is to obtain plants with enhanced properties. For example, increased content of secondary metabolites or amount of essential oils in medicinal and aromatic plants, or on the other hand, to obtain ornamental plants with more prominent parts, leaves, flowers, and so on. There are several breeding methods, one of which is in vitro induced polyploidization. This method uses several antimitotic agents like oryzolin and trichlorelin to induce polyploidization. Afterwards, plants are measured using chromosome counting, morphological and anatomical measurements, as well as flow cytometric measurements. Obtained plants would show different or changed morphological and chemical constituents compared to the plants they were derived from. The aim of this study was to obtain octoploid plants from the control of the original tetraploid Celozia argentea variety plumosa, which contains 36 chromosomes, using in vitro induced polyploidization. Our first hypothesis was that oryzolin is an effective antimitotic agent for the polyploidization of this plant, and then that the newly obtained genotypes of Celozia argentea would express different, hopefully enhanced morphological and biochemical properties. As for materials and methods, we obtained seeds of Celozia argentea from the botanical garden at the Faculty of Tropical Agri-Sciences. We used nodal segments from a single mature plant to maintain the genetical uniformity throughout the experiment. Let's look please at figure number 2. As we can see, the first picture has the mature plant in the Erlenmeyer flask that was removed, propagated, cultivated, where nodal segments as in identical clones of the mother plant were cultivated into different flasks that grew up, and this cultivation went on for 60 days on nutrient media until we obtained enough material to proceed with the experiment. The second part was the antimitotic agent, which we used, which is oryzolene. It has mitotic inhibitory effect, and we used it in 4 concentrations of 20, 40, 60, and 80 micromoles per liter. Every concentration was tested along 2 time intervals of 24 and 48 hours. If we look together at figure 3, the first picture here, we have the segments Celozia argentea propagated on the media, ready for the first variant. Here we're looking at variant 40 micromole per liter, so that concentration of oryzolene was added to this flask, the erylmine that we see in the first picture. And after exposure period of 24 hours, the segments were removed and washed for 3 times in distilled water. Eventually, each nodal segment was repropagated on the media clear of any growth regulators. The first result after the plants grew was to test if we have obtained polyploid or changed the number of chromosomes in the influence material. So we used float cytometry. It is a rapid and reliable method that shows the relative DNA content in plants. If we look at figure 4 on the left side, we see this. We see that the relative DNA content for the control plant here. If we look at the x-axis of the histogram, we see that this relative DNA content is showing at channel 200, and we already knew that this is the control plant. We repeated it several times to be sure, and then we moved on to measure the influenced plants to see if there is change. Let's go to the right side on figure 5. The histogram again on the x-axis. This time we see the relative DNA content showing on channel 400, which shows the positive or the polyploidization effect, where the chromosome number was doubled in the obtained or in this influenced plant. The second part of our results was to evaluate how much the antimitotic agent was effective. So here we look at this table at the survival rate and number of polyploid plants. We can look at our most effective variant, and this is 40 micromole concentration of oryzolene along exposure time of 24 hours. We obtained survival rate of 70.5%, from which 3 octoplate plants were obtained. In total, we used 320 nodal segments for all the experiment, from which 4 octoploids were obtained. Over here, we're looking at the morphological variation. We did this the morphological comparison 20 days after cultivation under in vitro conditions. So let's take a look together at the changes in the plant. If we look at part A on the left side, this is the control plant. We see that there are more shoots per plant, and the control plants produce the inflorescence way earlier than the influenced plant, the octoploid. On comparison, let's go now for the influenced plant, the octoploid. We're looking at the second picture B. We can see that clearly the stems are thicker, more compact growth overall for the plant, and most importantly, a very well developed root system. Of course, these are preliminary results. We are now still in the phase of continuing morphological measurements and chemical analysis to further see the changes in the new material. We know that polyploidization has different effects on many species and different families. Effects where morphological traits were changed. For example, a study done by Tan in 2000 and Collective in 2019 on Citrus riculata showed that the obtained tetraploid plants had larger fruits, larger seeds, larger leaf area index in the obtained tetraploids compared to the control diploids. And as well, polyploidization has an effect on biochemical profile of plants. In our recent study, we obtained an increased amount of essential oil in thyme, Thymus vulgaris, and also several components, secondary metabolites were increased. For example, thymol, which increased by 18% in the tetraploid plant compared to its diploid counterparts. Celozia argentea is really has been used for a long time in traditional medicine And there is high interest in this plant or for its medicinal potential these days. And several studies show that. For example, over here, Tang and Collective in 2016 isolated and identified 79 compounds of Celozia argentea, of which they state that saponins are the main pharmacological active agents. So of course, when we will obtain our chemical analysis, we hope, for example, that the saponins would have increased, or we would see if that would have happened on the expense of some other component and so on. Other studies, for example, they test the influence of physiological and developmental stage on the phytoconstituents and antioxidant potential of the plant. There are several also other agrotechnical studies like interrow spacing, watering systems, fertilizers that go for the horticultural aspect, which influence the quality of flower cuts. So of course, we also hope that the new genetic material would have had elevated or enhanced ornamental features. We concluded that oryzalane is an effective antimitotic agent where we obtained new octoplate plants for the first time. In vitro morphological studies are promising, and we will be doing already transferred the plants to ex vitro, and we will be doing the morphological analysis to obtain statistically significant differences for each morphological trait between control and octoploid plants, then between each octoploid. And simultaneously, of course, we will do the chemical analysis to identify changes in the chemical profile in the future as well. We can go for agronomic characteristics of the new material to see the biotic, abiotic stress, tolerance, eventually resistance of the plant, and so on. So this was our conclusion. This is the references that we used, and I thank you very much for your attention.
Yes, thank you very much for your nice presentation. Very well done, and also in time. I wonder whether someone in our room has a question. There's no question in the question and answer chat so far. Maybe to give others more time to think about their question, I can start with one. I was wondering whether the success rate, like 4 plants out of 320, would you say this is good or bad? And is there room for improvement?
I would say it is a good result because polyploidization is a process that goes through several procedures that go after each other. So there are certain variables that we always deal with, which are the explant type, and then the antimitotic agent of choice, the concentrations of that agent, and the time intervals that we would choose. Usually the survival rate is this low because in general the plant is going somehow kind of a stress factor which is causing the change. So according to other published studies, according to our previous work, yes, we consider, I would say that this is a good survival rate for the plant.
Okay, thank you. Yeah, congratulations for a good survival rate. Maybe my second question, I know you will still do the chemical analysis, but what is your hypothesis regarding the effect of the polyploidization on the nutritional quality and the medical quality of those plants?
The obtained genotypes are usually variable. We usually, it's more like an interesting result. When we come to measure the flow cytometry, we also have no idea how much plants we will obtain. After which, again, each genotype is specific and each genotype is different from the other. And overall, we cannot say how the result would be. For example, the first results, the morphology in the in vitro stage, they are promising for us to kind of expect that we might be looking at an enhanced plant. But this would go on after the ex vitro transfer, and the plant would perish, or it would be inferior to the diploid or the control from which it was induced from. This applies also to the chemical profile. The thing that is always observed in induced polyploidization of medicinal plants on the level of the biochemical profile is that always not all the components would increase and not all the components would decrease. Always something would increase, and that would be happening on the expense of another component. In certain studies, for example, the size of the vigor of the main part of the plant would increase, and that would happen at the expense of secondary metabolites. This would be explained that the plant here is using its the metabolism and the physiological energy is going for the more vigorous growth to deal maybe with biotic, abiotic stresses than it is to deal with for example, it also, if infected by a pathogen, then there's then the need to process more secondary metabolites to combat that. But yes, the result is always extremely variable. This is why we really look forward to see what has changed, and we hope that the compounds that are nowadays being studied and used in the pharmacological industry, we hope that these components would have increased.
Okay, so I wish you good luck with this.
Thank you very much.
Hopefully there will not be any dilution. But yeah, this needs to be proved. Okay, now here is a question in the chat.
Okay.
Richard, maybe would you like to turn on your micro and ask your question yourself? Or otherwise, I can also read it. Richard said, good presentation.
Thank you.
Oh, yes, Richard, go ahead.
Yeah, okay, thanks. Yeah, so that is a good presentation, my colleague there. So this is of genetics. I'm very keen on the plant you are working on, but I'm wondering what is it currently used for and how is it distributed in your country?
Okay, I am from Lebanon.
Yes.
The plant of choice was because at our faculty we are encouraged to choose plants from the tropical and tropical region, and our first author was from Vietnam. This is why the reason of choosing Celozia as the plant for the study. This is the main reason. Now the plant grows in the has a tropical origin, but it also grows in several countries. And the use dates long, but goes long back through traditional Chinese medicine. It has been used for several purposes. For example, it has anti-tumor function, which is really important. Then also if I'm going to mention something from the traditional medicine that was from which, of course, this was inspired, that it was used in therapeutic effect and for treating eye diseases. And this is one thing that it was already used in traditional medicine, and actually the pharmaceutical industry went on to identify exactly which compounds are responsible for this effect. The plant also has antioxidant effects and anti-infectious activity.
Thank you very much. Thanks for your elaborate answer.
Yes, thank you also for the question. I think not everybody would know this plant. I know that for example, in the Andes in Peru, amaranthus species are also very common and a very good contribution to the people's nutrition. Yeah, so nice. So now we can see we have 10 minutes left. Are there further questions to Yamen, or we could open up more generally? All right, now I don't see any other question. Maybe to use this time, well, I would like to give you again the floor and maybe summarize in 1 or 2 minutes how your work actually contributes to the overall theme of this conference, namely food and nutrition security and its resilience to global crisis. And so I would, each of you could make a final statement how your study contributes to food and nutrition security and resilience in global crisis. And we would, so starting with Aslan, then Joshua, Richard, and then Yamen. Sorry to put you a little bit on the spot, Aslan. Maybe you, and you could stop sharing the screen, Yamen. So Aslan, would you be ready to summarize how your study actually contributes to food and nutrition security and resilience? Please open up your microphone. Are you still there? Aslan, are you still there? Please open your microphone. You are still muted. Well, if he's not ready, maybe who would be ready to make a final statement for this session?
I can go. I can.
Okay, Joshua. Yes, please go ahead. Great.
Yeah. So thanks very much for this reflection about how my work can contribute to the general theme of the conference of food and nutrition security. I think sweet potato is a food security crop. Almost every household in Uganda grows it. So, and it matures quickly, and it requires very few inputs to grow. Even the poorest household can grow. So it's really key. It can contribute significantly to food security. It can be harvested in case the yield is big. It can be harvested and stored for a long time. You can store it in sand, or you can process it in flour and store it, keep it for a long time, or you can use it as an ingredient in other bakery products like we learned. And when it comes to resilience in the changing, in the global crisis, for example, of the COVID-19, if that's what I would take, or in the climate change aspect, is that it's quite drought tolerant. It can grow even in arid areas with little water. So it's quite, then it's being nutritious, like having the component of vitamin A that boosts body immunity. So it is what we find it as being very important in this era where you need to have people need to eat nutritiously and boost their body immunities. We mainly work also in the fragile environments where there are refugees coming in, and refugees have small plots of land, so they can use this relatively small plots of land like in the home gardens, and they're able to produce quite sufficient amount of food. The yield is relatively good. You can get up to 30, 40 tons per hectare. So we feel that sweet potato is really very, very key in resilience. Like you can also do piecemeal harvesting. You don't have to harvest at once when the crop is mature because roots form at different times. So you can go on picking the mature roots, the bigger roots, as you eat and as the other roots continue to grow. So that's why I feel that the component or using the orange-free sweet potato is it fits in very well in the theme of this conference.
Okay, I think this was quite convincing. Thank you very much. Now, who else would be ready to make a final statement?
I would.
Maybe I
yes,
can.
Yaman. Okay, Yaman and then Richard.
Okay, so nowadays the usage of secondary metabolites that are naturally occurring constituents in plants show to be a sustainable form of combating diseases and medication, as well as in the agricultural sector, instead of using commercially synthesized chemicals. So here is where the effect of these plants, and exactly in our study what we are doing is that with the increased interest in these plants, the natural need and the picking from the wild of these plants, as well as supplying the supply for the commercial production. So this work comes in turn by increasing the content of these metabolites per plant to address the increased demand on medicinal and aromatic plants. Thank you. This is it.
Thank you. Yes. Also a very important future. So now, Richard, I give you the.
Yeah, thank you. So the way my study will contribute to the overall theme of the conference, first and foremost, we know chickpea is a very nutritious crop with multiple uses. And again, as I mentioned, that the chickpea is able to grow under marginal conditions. In the advent of climate change, we talk of resilience, and by virtue of the fact that it grows under marginal conditions, then it means it can be grown off season, and therefore, and again from the perspective of being nutritious, it can be grown almost throughout the year, and therefore contributing to food security and also nutrition and resilience by virtue of the fact that it is used as a mitigation strategy to climate change. I think that's what I can mention in relation to my project, to the overall theme of the conference.
Yes, thank you very much. I think legume important future and an important contribution to resilience in any case, and also nutrition. Mr. Aslan, if you are still there, you could also still make a final statement. Right now, you are still muted. There, or can't hear us, or can't unmute. Just thank you very much for this nice sessions and convincing contributions to food and nutrition security and resilience in a global crisis. Thank you very much. I'd like to close the session. Okay.
Thank
Bye.
you, Bettina. Bye.
Thank you. Yeah. Bye-bye.